Okay, so today we're going to learn about selective and differential media. These are specialized media that are very useful in helping to identify characteristics of an unknown bacteria. The three we are looking at today are phenyl ethyl alcohol agar, which is abbreviated PEA, mannitol salt agar, which is abbreviated MSA, McConkey agar, which is abbreviated MAC, and EMB, which is eosinethylene blue agar, so it's abbreviated EMB. Okay, so the PEA looks a lot like a nutrient agar, so you guys want to make sure you label this plate so somebody knows it's not nutrient agar, including yourself. Okay, the MSA, mannitol salt agar, is kind of a reddish color, a little bit pinky red. The McConkey is a light brick red color. And the EMB is a nice purpley color. These are the colors they are when they start out. After they've incubated, some of these medias change color, sometimes depending on the bacteria that's on there, and sometimes just because it's in the incubator and it's been at 37 degrees Celsius. So make sure you label these plates before you put them in the incubator so you know which one is which, because they won't necessarily look like this when they come out of the incubator. Okay, so there is a handout on Blackboard that goes through each of these medias, okay? And we're going to be inoculating them with 4 different bacteria. So we're going to need a plate of each for each bacteria. All right, but I'm going to go over the medias themselves in a little more detail right now. So if you go to the second page, you will see that the first one it goes over is PEA, phenyl ethyl alcohol agar. So that's the one that looks a lot like nutrient agar. This particular media is only selective. It is not selective and differential. Okay, so what does that mean? All right, so there's selective ingredients in all 4 of these media. The selective ingredient will either allow growth or not allow growth. Okay, so it's a yes or no answer. There's growth or there's no growth. Okay, and just that answer can tell you a lot. Okay, which we'll learn in these tables. But the selective ingredient is what gives that media the ability to do that. So in this case, the selective ingredient is phenyl ethyl alcohol agar, or alcohol. So it's in this agar, and it will either allow growth or not allow growth, depending on the bacterial characteristics. Okay, now the other 3 are selective and differential. Okay, that means that on top of a selective ingredient, for instance, MSA, the selective ingredient is NaCl, okay, salt. That ingredient will either allow the bacteria to grow or not grow, and that will tell us something, which we'll go into. And on top of that, if the bacteria grows, and only if it grows, the differential ingredient, which is, in this case, phenol red, will allow you to tell more about that particular bacteria, okay? So the PEA is a selective media only. Okay, selective means it allows growth or it doesn't allow growth. Okay, so it's giving you a simple answer. For instance, it could be gram positive versus gram negative. Okay, so that's what that is telling you. The differential ingredients in these 3 other media, okay, they're selective and differential, will allow you to tell more information about each of the unknown bacteria if they grow, but only if they grow. Okay, so for instance, to give more details, your PEA, okay, it tells you in your handout. This is very important information. It should be in your notebook so that you can reference it very quickly. It's going to be on quizzes and exams. It's going to be prevalent on both of those, that you'll be able to access this information, read it, and be able to answer questions quickly. So make sure you go over this, make sure you put it in your notebook, okay, you're familiar with it. So the PEA agar. This is only a selective media, so it's got a real short table and a short explanation. So it tells you right at the top, it selects for gram-positive organisms, meaning it selects for, meaning it allows them to grow. Okay, so the selective ingredient you'll see is phenyl ethyl alcohol. If the bacteria can grow, it is not inhibited. Okay, it's being allowed to grow. It's not inhibited by the phenyl ethyl alcohol. Okay, and this will tell you then that it's gram-positive. Okay, and it tells you that in this table. Okay, result. The poor growth or no growth at the top, Okay, if there's poor growth or no growth on here, it's telling you to interpret it as it is inhibited by NaCl. Okay, it cannot grow on there, so it's inhibited by NaCl. And this tells you that it is a gram-negative bacteria. Okay? Now, if it allows growth, okay, because remember it says PEA selects for gram-positives. If it allows growth, it's not inhibited by the phenylethyl alcohol. Okay? That means that it is gram-positive because that phenylethyl alcohol did not inhibit it. Okay, so you have a gram-positive organism. So that's outlined in this table. Okay, so that's the PEA, which is just a selective media, so that was a little more simple. I want to go down to MSA so that we go over a selective and differential media also. So MSA, which is mannitol salt agar, you'll see that it says it selects for Staphylococcus species. Okay, so that is what MSA is primarily used for. It also tells you the selective ingredient is salt, okay, 7.5% NaCl. Okay, so the selective ingredient is either allowing growth or not allowing growth, remember. And then it goes on to tell you that the differential ingredient, because remember there's a differential ingredient here that gives you more information. The differential ingredient is phenol red. Okay, that's a pH indicator. It'll turn yellow if there is acidic conditions. Okay, so the pH goes into an acidic area on this plate. Okay, if the bacteria is producing acid, it will turn this media yellow from red. Okay, so we have an unknown bacteria. We put it on this MSA plate, and then once we have incubated it, we're going to read the results. Okay, so this one's a little more complicated of a table. So the results could be poor growth or no growth. Okay, so say I put E. coli on here, okay, and I incubated it, I look at it, there's no growth on it. Okay, the E. coli did not grow. So that is telling you, okay, the interpretation that the organism is inhibited by salt. Okay, it cannot grow on the NaCl. So it's inhibited by the selective ingredient. So that tells you that it is not a Staphylococcus species, which makes sense because you put E. coli in here. Okay, so it shouldn't have grown. It should have been inhibited by NaCl. It's not a Staphylococcus species. Okay, so in that case, you can't tell anything else because nothing grew on this plate. Okay, so your results for that plate that has no growth on it would be across here, letter A, poor growth or no growth. The organism is inhibited by NaCl, and it is not a Staphylococcus species. Okay? Now, if there was growth, if there's growth, good growth, you'll see under B, good growth. Okay, that tells you something right there. Good growth tells you something. It tells you, first of all, that the organism is not inhibited by NaCl because it can grow. Okay, so it's not inhibited by NaCl. So that right there tells you that it is a Staphylococcus species. Okay, so you've already determined something just from growth or no growth, right? If there's no growth, it's not a Staphylococcus species and it's inhibited by NaCl. If there is growth, it is not inhibited by NaCl and it is a Staphylococcus species. Okay? Now, that's the selective part of it. But now that it grew, you can also tell the differential characteristics because this is a selective and differential media. So its differential ingredient is phenol red. Okay, and this agar also has mannitol, and it is sugar. Okay, so B was true, good growth. You can tell this row of information in B. And B also says, if this is true, it's also C or D also. Okay, those are your differential characteristics. So C says the media turned yellow around the growth. So say we put an unknown organism on here, and it grew, and the media turned yellow. And it's a really obvious yellow. There's some pictures of some examples, accompanying the handout on Blackboard. So the media turned yellow. Okay, so that bacteria, when it was growing, created an acidic environment, caused that media to turn yellow. Okay, so that tells you that the organism produces acid from fermenting the mannitol, the sugar that's in here. Okay, so that tells you, as a presumptive ID, it is a possible pathogenic Staphylococcus aureus, okay, which can be important because that can be a pathogen. It could be a problem in a wound, or if somebody has staph poisoning or something like that. Okay, so that would tell you that that's Staphylococcus aureus most likely. Now, if there's good growth, but the media did not turn yellow, that means that it did not ferment the mannitol. Okay, so you'll see down at the bottom, media remains red around the growth. Okay, that means the organism does not ferment mannitol. Okay, so it's not producing that acid, it's not turning the MSA agar yellow, it remains red, okay, because it cannot ferment the mannitol. And that tells you that it's a non-pathogenic staphylococcus, such as Staphylococcus epidermidis, which is very common on our skin, under our fingernails, everywhere else. Okay, so that tells you that this is a non-pathogenic Staphylococcus, or Staphylococcus species. All right, so when you have growth on it, remember you can tell this, okay, you can tell it's not inhibited by NaCl, you can tell that it's a Staphylococcus species, and then from there you can tell additional information because of the differential ingredient, phenol red. You can tell if it is producing acid from mannitol fermentation by turning the media yellow, okay, and then that will tell you it's a possible pathogenic Staphylococcus aureus, or it can tell you that it cannot ferment mannitol because the media remains red, and that will tell you that it is a non-pathogenic Staphylococcus aureus, okay. And then this handout goes on to explain the Conky agar in the same manner, and EMB in the same manner, okay, there's tables for each of them. So you should make sure you familiarize yourself with these tables. You're going to be using them to read the results on plates in the future, especially on quizzes and lab exams. You want to be able to reference this information quickly and understand what it means. Okay, so today we are going to be using 4 different bacteria. You'll see them listed. So we got E. coli, Proteus, Vulgaris, Staphylococcus aureus, and Staphylococcus epidermidis. Okay, we're using those 4 bacteria to show how the selective and differential medias work, and so that you can see what different outcomes of results would look like. So that if you had an unknown bacteria, you would know what you were reading, what kind of results you were reading. I'm going to go through just one inoculation of one bacteria because it's just repetitious of the other 3. So I will pick the E. coli. Okay, I'll put those 3 aside. So I've got the E. coli culture. Remember, it's a broth culture. You want to agitate it back and forth. Don't shake it up and down, back and forth, so you suspend the cells. Okay, and you have one of each of the media, and you have labeled them with the type of media, very important. Remember, they can change color once they go in the incubator. The organism that you're putting on it, which in this case is E. coli, and the date and your initials. Okay, so you're going to do that with each. So this is PEA, this is MSA, this is McConkey, MAC, and this is EMB. Okay, and it also has E. coli written on each of these, and my initials and the date. Okay, so today we are using sterile swabs to inoculate, just for the ease of application. We're not trying to get isolated colonies or anything like that, so we're just going to use a sterile swab. The sterile swabs, they open like a Band-Aid, so it's got a little area up here you can pull apart. If you can't find the top where you should peel it apart, feel where the wood stick is. Okay, that's what you want to open. You don't want to open it at the swab end because you got to have that remain sterile when you're putting that into your bacteria. You want to get your fingers all over it. Okay, so you have all your plates labeled and ready to go. Okay, you're going to need your Bunsen burner lit. So go ahead and light that. Okay, and you can peel this down to get ready, just so you have the stick sticking out. Your swab is still down here, nice and sterile. Okay, you have your plates all ready to go. You have your culture. So you're going to pick it up. Okay, agitate it. Go ahead and get your swab. Okay, pull your swab out. So now you're ready. You do not flame this. Obviously, it'll set it on fire, so don't do that. Okay, it is sterile already. Pick up your tube of bacteria. In this case, it's the E. coli. Remove your lid. Remember, use your little finger. Flame the top. Okay, you don't have to cool this because it's cool. Go in and soak that swab in the bacteria. Come out. Flame the top. Put the lid on. Put the E. coli down. Okay, and we'll take the first plate, which is our PEA plate. Remember, we use it lid down. And we're just going to pick it up and just gently take that swab and go back and forth across the surface of the agar without digging it in. Okay, so we'll put that plate down. It's been inoculated. Okay, and then we're going to pick up our culture again. We can reuse this swab because we're using the same bacteria. Okay, so it doesn't matter that there's E. coli already on this because we're going to go back into the E. coli. But we do have to flame the top of our tube each time. So we're going to remove the lid, flame the top, take that swab, go in, soak the swab, flame the top, put the lid on, and then pick up our MSA plate and gently swab back and forth against the surface of an MSA plate. Put that down. It's been inoculated. Remember, don't put this in the flame. Don't put it down either, because then it will be contaminated. Pick up your tube again. Remove the lid. Clean the top. Go in. Soak your swab. Flame the top. Put it down. Pick up your conky plate. Look at and gently swipe it across the surface of the mud hockey plate. We're done with that one. Lastly, we're going to pick the tube up again. Flame the top. Go in. Get some bacteria on your swab. Flame the top. Put it down. And then go ahead and inoculate your last plate, which is the EMB, by swiping back and forth across the outer. Okay, now you're finished with this swab. You're finished with all your plates. So what you do is gently stick this back in your wrapper. And then you dispose of it in something that, some part, container that's going to be autoclaved, okay, because you want to kill the bacteria in here. So you never put this in your regular trash. Okay, so this is a little disposal bin that has contaminated items in it that I will, now that it's full, take it, put in the autoclave, kill everything before it goes into the regular trash. Okay, so we have successfully inoculated each of these 4 selective and direct differential medias with E. coli. Okay, so we will put those in the incubator, okay, for 24 to 48 hours at 37 degrees Celsius. We will get another 4, okay, one of each of the selective and differential medias, and then we'll do the next bacteria, which is Proteus vulgaris. Okay, and then we'll get another set for our Staphylococcus aureus, and another set of plates for our Staphylococcus epidermidis. So in the end, we're going to have 4 plates, selective differential media for each of the 4 organisms. And then we'll be reading the results of those plates, which will be posted so that you can see what they look like. And that ends this lesson. Thank you. Actually, turn off your lesson.